So I would like to open the presidential lecture. So today we have our excellent guest speakers from Italy. And while he's coming up to the podium, I would like to briefly introduce Professor Rizzolatti. Professor Giacomo Rizzolatti graduated from University of Padua in Italy in 1961. And he also received training in neurology, physiology at the University of Pisa, and psychology at the McMaster University in Canada. And most of his scientific career has been done at the University of Parma, where he is serving as a professor emeritus of human physiology. And he has been visiting professor for many universities around the world, including University of Pennsylvania and Philadelphia, or some other university, University of California, Santa Barbara. And he is a president, he has been the president of European Brain Behavioral Society and Italian Society of Neuroscience and many others. And he also a member of many domestic and international societies, too many to mention. So, and he received so many awards, including Golgi Award for Neurophysiology, and it takes an hour to introduce all of them. So he's very famous in his work on cognitive function, on motor neuron system, and discovery of the mirror mechanism. And he has written many scientific articles on this topic. And he also proposed the premotor theory of attention as well. So he has more than 500 international publications, and they were cited 150,000 times. And he has H-index of 142, so big number. The title of his talk today is From Mirror Neurons to Mirror Brains. So, Dr. Rizzolatti, please start your lecture.
Well, thank you very much for... Can you hear me? Thank you very much for your presentation. I am really very happy to be here. I was a bit surprised when Professor Rizzolatti told me that I have been invited to give this lecture, because I am a neurologist, a neuroscientist, but know nothing about spinal cord. If you go away with the next slide, here is the person which I want to thank. First of all, Professor Kosumi, and then my friend Negrini and Zaimi. Well, when I received this invitation, I was really very perplexed. Then I read, next slide please. Let us remember that importance of collaboration, open dialogue, and the pursuit of excellence, it's one of the fundamental points of this society. And when I read that, I say, yes, I must come. It's interesting. So my talk will be essentially on the mirror system, and then what we are doing now, which is essentially a rehabilitation. We apply the mirror mechanism to rehabilitation. The next slide shows you the beginning of the story. At the end of last century, still the neurologists and neuroscientists thought that there are two motor areas, area 4 and area 6. Area 4 and area 6. But the people were not very happy with this very simple subdivision. And in Parma, we start to do, this brain is a monkey brain. We start to do a series of histological data, and we found that indeed area 6, the Brodmann area 6, is formed by several areas. I am interested in one of them, which is called area 5, F5. We study it using single neuron technique, which was very popular in those days. And that was, I think, our great idea to use an ethological approach. Typically, the motor system is studied in terms of kinematics, size of the object, force, and so on. We started to do ethological approach. So we were in front of the monkey, we gave to the monkey a piece of food, we take away from it, and so on. And the results were surprising. The first one, if you look at the left part of this picture, forget the right part, it's the right hand grasping an object. And in the lower part, it's again some movement of the fingers. So in both cases, there are movements, but in one case, there is a goal grasping, in the other not. So we make this distinction between movement and motor acts. Movement are simply displacement of a joint. Motor act, well, there is a goal. Incidentally, this part of experiment has been carried out in collaboration with Nihon University in Japan, and in particular with Hideo Sakata and Akira Murata. Well, the results are the following, that if we are true, and it's indeed what it's called in this area, is the act, the motor act, the goal, these data you see here are easy to explain. The monkey is grasping with the mouth, the monkey is grasping with the right hand, with the left hand. But the goal is always the same. Remember that, the goal, not the movement. It's the goal which is very important. The big surprise was what is here. If you look in the lower part, B, the monkey is grasping an object, and you see the action potential. All these lines are action potential in different trials. But look at A. There is a person grasping, and yet there is a discharge. The discharge is very similar. And now I would like to show you a movie in which you will see, you will hear the action potential, tac, 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 and you will correlate this tac, 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 tac with the action of the monkey. Please, the next one. The next one. I think that, okay. Can you? Here is the monkey grasping an object, and it tac, tac, tac that you hear are action potentials.
Prima, feeling, ancora.
It's highly repeatable. And here is the mirror effect. It's the same neuron which fire in both conditions. It's like a mirror. It's the same action done by the experimenter and by the monkey. And this one, you know why. It's grasping with the mouth. Now, I am a neurologist. I am doing experiment on monkey, especially in the past. But of course, what is interesting is what happened in humans. And we were so happy that in that time, in San Raffaele in Milano, there was a pet center just founded in those years. The director was Ferruccio Fazio. I was a good friend of Daniela Perani. So we did an experiment in humans. The pet experiment in humans was done like in the monkey. The person was lying in the scanner, and we injected a substance. The substance was desoxyglucose, radioactive. So the part of the brain which is more active capture more desoxyglucose, and so there is more radioactivity. And the result were multi. You see, this is a human brain. The yellow stuff corresponds to F5, to ventral part of F6, and the red part, we didn't know at the time, we discovered it in humans, that also the parietal lobe have mirror neurons. Here was a problem that students don't want to be injected with a substance containing radioactivity. So it was very difficult to find, for the patient, obviously, the patient said, well, it's for me, I accept radioactivity. But the patient said, no, why? Maybe then I will have a cancer. Or fortunately, some years later appeared fMRI. Now probably all of you know what is fMRI. It's all hospitals have fMRI. And we used fMRI. This was an enormous success because many labs in the whole world used fMRI to study mirror neurons. And here is a review made in Germany, in Jülich. The yellow stuff is what happens when a person sees, observes, not that, an action. You see there is an activity in the occipital lobe, posterior part yellow, then in the parietal lobe and in the frontal lobe. That was done on 100 cases. So it's really very rich. Now there is even more than that. Why? At the beginning, we thought imitation, and it's reasonable to think imitation. But then ethologists told us, listen, everybody thinks that the monkey imitates, but the monkey does not imitate. The monkey can make a grimace, can make something similar, but they are unable to learn by imitation. And in the meantime, other people found mirror mechanism in other species, like, for example, in bat, in rat, in mouses, and so on. So it's not imitation. What could be? The other idea was action understanding. So when you see somebody doing an action, you understand it because inside your brain there is the same action. Imagine that you go in a coffee and see somebody grasping a cup of coffee. Well, inside of your brain there is also the motor program for grasping a cup of coffee. However, we had some friend who said, no, it's impossible, because I understand the fly of a bird, and I have no mirror neurons for flying. Well, we did an experiment, fMRI experiment, and we presented to a student these films. In the first, you have a boy grasping with the mouth a piece of food. In the second is the monkey doing the same, and the third is a dog. Well, the results showed that all part of the brain which became active when you grasp, person, human being, grasping, became active when also the animal grasping. The things, however, are completely different here. Here you see actions which are typical of a species. A human being was observing a person talking. The monkey was doing a typically ethological gesture. It's a kind of, let's be friends, we are friends, don't go do the war. And then the dog. Look at the activity in the dog. In the dog, there is nothing motor. So we understand that the dog is barking, and all students who have been asked what they saw. Why? Because there are two ways, we discussed it with several philosophers, because there are two different ways in which we understand action of others. One is understanding from the inside. As I said before, I see somebody grasping a cup of coffee, and I'm grasping the cup of coffee. Inside of myself, the motor program is grasping. But for something, no. For mathematics, no. For flying the birds, flying the flight, we have to think. There is a cognitive way to understand it. Well, mirror neurons had enormous success. Then at a certain point, there was a decaying fact. Fortunately, what happens is that we and others discovered that showing action observation can be useful as a rehabilitation. Here it's perception of other action can trigger, well, it doesn't matter. You can read it. Or can you read
Okay,
it?
perception of another person's action can trigger the same action in the observer because perceptual event representations and representations of actions share a common representational domain. I don't understand, but I can read.
Thank you. Well, we started this experiment of rehabilitation in Parma. We started the rehabilitation in Parma at the beginning of century. And while both doctor and technician were very helpful, the administration was not very happy. Why we have to call back patient and so on. So the first experiment, although we are involved, has been done in Germany, in Hamburg, by Erfeld. And he wrote, action observation has a positive impact on rehabilitation of motor deficits. Subsequently, in Bellelli is an orthopedics, and he works with many others. It's okay, you can see it. Thank you. And he showed it. No, I see very well now.
Do you want me to read? No, you can. Yeah, it's working.
It's okay.
Yeah, it's okay.
So in orthopedics, it was very, very successful because the rehabilitation of person who has been blocked in this position, for example, for a long time, recovered in 1 week. And in orthopedics now it's very, very commonly used. Thank you. That's during COVID with my coworker, we decided to write a review, and we showed that the mirror mechanism is important in the recovery, maintenance, and acquisition of motor abilities. And now it's a kind of gigantic quantity of people who work in that. I quoted here some of them. Stroke, which is one of the first used, children with cerebral palsy, Parkinson's disease. In Genova, there is a group who worked a lot on Parkinson's disease, multiple sclerosis, and as a neurologist, I was first surprised at why, and also in hip prosthesis. You can say why this happens, because if I have a blocked my arm, the motor center which control this tend to disappear, become weak. And when you have free arm, your capacity to move is not good. Instead, if you observe it, the motor system, the central motor system was active also if you were not moving. And when you put away the cast or what it is, the movement is much better. And that's true for the other thing. For example, for multiple sclerosis, there are phases in which the person cannot move, but if in that period you show a correct movement, in the moment in which you put away which he recovers, there is again a good movement. I have still 10 minutes. I want to say to something which also had a great success, emotions. These are, when I talk of emotion, I speak about this basic emotion, anger, fear, surprise, and so on. And we studied disgust. Somebody could be surprised, but disgust, according to Darwin, is the most basic emotion that we have. So we did an experiment, which is shown here, showing to the person, a guy, a gentleman disgusted, a gentleman which show pleasure, and a neutral one. And the activity was in the rostral part of the insula. People in London before us showed that exactly that part of the insula became active if you gave bad odorants. We repeated the experiment, we also have that. And look here is the most important thing. There are voxels which are white. The white voxels are those voxels which became active both when you are disgusted because you received a disgusting stimulus and when you see a person showing disgust. Note that these individuals were absolutely pleasant, was not disgusting at all. And that's exactly what is empathy. Empathy is that part of the brain which became active both when you are disgusted and he is disgusted. You feel pain and the other person is pain. More recently, and that's the end of my talk, we collaborated with a center in Milano, Niguarda, which is working on epilepsy. Epilepsy is a disease which fortunately is cured with drugs, but there is a group, the people which are not responsive to drugs. So we worked with them, and it's a very expensive experiment because you must have fMRI, you must have a special electrode, you must do arteriography on the left, you must have insert. So they insert in the brain of a patient many electrodes, and for 2 weeks the patient is in the hospital. They record if they are epileptic crisis, both using video and using EEG. Then they came to us and saying, listen, we have a lot of material because here you see in this white brain, there is many electrodes and the epilepsy will be only one part of the brain, but there is all this material that we can work together, understand what it's doing it. So we use 2 systems, intracranial stimulation and recording. I have no time, so I will tell you what happens with intracranial stimulation. That's important because remember that with fMRI, you have a correlation. You don't know if really this area is active. You know that this area became active during, but you know if it's responsible. Here it's the mesial part of the brain with different area, and I will show you only 2 experiments. One is the stimulation of the blue part, that called MIA CVV. Please.
1, 2, 3, 4, 5, 6.
Che c'è? Cosa è successo?
Tu vuoi andare via?
Cioè?
Hai sentito che vuoi andare via così?
So he said, I cannot stay. I don't know why, but I have to go away. I cannot stay here. And that has been repeated in other lab. I wanted to go away. look that psychologists interpreted the stimulation of this area in many ways, like pain, different type of stress and so on. Actually, these results show that these different topics that they described, it's anxiety. This is an area which when you stimulate, the person became anxious. And here you see the anxiety. I have another case to show you. In this case, we stimulate the red area. You see the red area in the front. Here is a girl. Go ahead. I can go.
Lamuro coltello, maestra, ferro, gatto, arancio, pulsante alto, orecchino. No, è un attimo di...
So the instruction was, tell this word, she said, orange and so on. At a certain point, she started laughing. If you think you are a patient, you must be operated in few days, you are in the hospital, at a certain point you start laughing. Why laughing? Another case, here is a boy, prego, fa partire.
Allora, mi dice il suo nome e cognome, data di nascita. Cosa ride?
It's the same area, and the next one, this one.
Zanzara, foglie, pane, schermo, borsa, albero, nuvola, cancello, laccio, acqua. Zanzara, foglio, pane, schermo, borsa, albero, nuvola, cancello, laccio, acqua. Non mi viene fuori. Non sono
He
io.
cannot stop, and he said, it's coming out of me, but it's not me. So it's not him, it's not his frontal lobe, his will, that's saying laugh. But the part of the brain we stimulate compel him laughing. The last slide was our mirror neurons. So we presented to the same patient in which we know where is the area which provoke laughing, films, and they saw a video in which the boy is crying, in which is laughing, and a neutral situation. And if you look at the left, the red light indicates the activity of the brain when he sees laughing. So exactly the same area which whose stimulation produce laughing is the area which is determined laughing when we see it. Empathy. This is definition of empathy in German, but it's easy to understand. It's the relation that stresses the mutual holistic existence of two beings. It is a concrete encounter between these beings, meet one with the other. In other words, empathy is when you and I are in the same condition. It's not simply that I am sorry because there is a war in Palestine. That's not empathy, that's compassion, it's what you want. But the real empathy, when you and I, and that's very important for the doctor has to understand the condition of the patient. I think empathy is one of the basic properties for a good doctor. The end, I am finished.
Thank you very much, Dr. Rizzolatti, Professor Rizzolatti. Very interesting talk, but too difficult for me to understand some part. But thank you very much for letting me be part of your presentation. It was very honored for me. So now, I asked Professor Rizzolatti, we have Lake Como tour this afternoon, so please stay in time. So he was so kind enough to, left us time for questions. So please come up to the microphone if you have any questions. No questions at all? Oh, yes, please. Ed.
Thank you. This was so interesting. I was wondering, as I'm observing my first grandchild, that she watches us do something. Oddly, watches us climb upstairs. And when we climb upstairs, we take one step at a time. And that little girl, who's 13 months, when we take her over to the stairs, she tries to take each step one at a time. Is that mirroring? Is she learning from observing? And is that part of what you're talking about when you talk about the mirror brain? Thank you.
So your question is the mirror system is involved in this imitation? What?
Yes.
I think so. So my point is that there is part of the mirror system which is innate. For example, the mother is a boy is crying, the mother understands crying, even when the boy is very small, it's an infant. With some other things you learn. So for example, walking, imitation of walking is something which it's learned. It's mirror neurons which develops in the person progressively.
Thank you.
So,
Thank you so much for all your research. I was wondering if you ever searched anything that has to do somehow with the trunk, because the movements of the arms are linked much more to the voluntary movements, while the spine and trunk is before we act with the arms. So it's really so basic that when we have to learn and we have to teach our patients some movements, or is going much deeper than just the voluntary movement. Is something that comes before, even before the voluntary movement. So you showed us the movement of the arms related to the goal, and again, the spine is not directly related to the goal, because the goal is done with the arms, and the spine stabilizes to allow us to move. You showed about the emotions, so I was wondering, and there are two totally different things. One is the active movement, the other one is the emotion. I was wondering what could be, if you did something about the trunk, you explored something that could help us treating our patients. What was clear?
If I understood correctly, you somehow separated motion from emotion, or that was bad microphone. Because I will separate completely emotion from motion. Motion is something that have no emotional content. There is nothing emotion in that. If I want to do that against our president, it will be full of emotion. So, but what we plan is that we have a motor program. That's the beginning of our experiment. Motor program for simple things like grasping, moving. And we have evidence, because the same neuron which fire when the monkey grasps, fire when it sees you grasping it. Emotion, I think, is completely another story, and it's more related to other centers like insula, amygdala, cingulate, and so on. Again, there is a mirroring between what you are doing and what I am feeling, but it's another field, because when I feel an emotion, I have also a visceral response, my heart increases, when I mentioned the war and saying it's not exactly like empathy, if I go out here and see an accident, immediately I have an emotional reaction, increasing vegetative responses, heart and so on. If I read that there were some dead person in the war, I am sorry, but if you measure my vegetative responses, nothing happened. I don't know if I not completely answered, but I don't know, what do you mean by motion and emotion? You want to separate these two fields?
No, I was separating the movement of the arms from the function of the trunk and spine. The function of the trunk and spine is before the voluntary movement. We stabilize and then we move. And so I was asking if you had any kind of results that related somehow directly to the spine and the trunk. That was my question. But I don't think...
I have not studied other action like walking and so on, but there are data from other labs which indicate that in supplementary motor area, for example, the same neuron which fire when you walk, fire also when you see somebody walking or moving the arm in a walking way.
Okay. Francis, please.
Thank you for a wonderful talk.
Last one. Okay, please, the last one also, because I can hear not very well.
Thank you for a wonderful talk. I wanted to ask if you could speculate how you would design your experiments today, given that so much of our interaction is not face-to-face, but through screens. Do you think it will have an impact on the way children, for example, learn and mirror?
Can you?
Francis, can you repeat your questions again?
Would you set up your experiments differently now?
Senti, ferma questa discussione perché That's an interesting question, but there are experiments made in Japan which show there is a very strong difference between what you see in a movie and what you see in a theater, not in real life, but in theater. You know that it's false. And it's much more strong, the effect, when you see the real action in relation to movie. You can use it because, for example, many of these experiments I mentioned were bidimensional. Now we are doing three-dimensional picture. But the real life is different from a picture of life.
Thank you very much.
Thank you very much. So please give a big hand to Professor Rizzolatti once again.